Good evening. On behalf of the National Stem Cell Foundation of Australia, welcome to Future Medicine, Stem Cells and Brain Health. My name is Tanya Ha. I'm Director of Engagement at Science in Public, where I've looked after communication for the Stem Cell Foundation for about 12 years, and I'm delighted to be your host this evening. I'd like to begin by acknowledging the traditional owners of the lands from which we are each joining this event. I personally am coming to you from the lands of the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past and present and recognise any people from other First Nations who might be joining us this evening. And we will be using the chat box to ask questions later tonight. So to exercise your ability to use the chat box in Zoom, why don't you tell us whose lands you're joining us from? On to the topic of this evening. The human brain is a wonderful thing. It's the seat of your individuality, your personality, and your abilities, for better or worse. It's about 2% of your body weight, but consumes 20% of your body's energy needs. Now, whales, dogs, lizards, frogs, and even fish have brains, but ours can solve Wordle puzzles. It can memorize Taylor Swift lyrics and coordinate tennis serves. Well, it can attempt to coordinate tennis serves in my case. But our brains are vulnerable to injury and aging. About 2 in 5 Australians will experience a brain disorder in their lifetime, and we still have so much to learn about the brain and what happens as we age. The National Stem Cell Foundation of Australia is a registered health promotion charity with a mission to promote the study and use of stem cells in the prevention or control of diseases, to alleviate human suffering, and to enhance public education in this field, which is why we're holding this webinar tonight. We're going to hear from 3 top scientists paving the way for new therapies and a better understanding of conditions that affect our brain and nervous systems. In short, we're going to pick the brains of top brain experts. They are Professor Carolyn Sue, AM, a clinician scientist at Neuroscience Research Australia, or NEURA, in Sydney, who researches Parkinson's disease and mitochondrial disorders. Professor Claire Parrish from the Florey in Melbourne, who is investigating cell therapies for Parkinson's disease and other neurological conditions, including stroke, motor neuron disease, and Huntington's disease. And our third speaker is Professor Kayleen Young from the Menzies Institute for Medical Research at the University of Tasmania in Hobart, who researches multiple sclerosis and other neurological conditions. I'll be talking to them about their research and what we can reasonably hope for in the future. Later, you will have the chance to ask your own questions. Please write them in the Zoom chat box and we will get to as many as we possibly can. Now, behind the Kieran Top Science in Public participant is my colleague Kieran. He is providing technical support, so you can send technical issues to him directly via the chat function. And do note that this evening's event is being recorded so that we can share it online at a later date. Now, it gives me great pleasure to introduce our first speaker, Professor Carolyn Sue, a clinician scientist renowned for her pioneering research in Parkinson's disease and mitochondrial disorders. She holds the Kinghorn Chair of Neurodegeneration at Neuroscience Research Australia, is a professor of neurology at the University of New South Wales, and is Director of Neurosciences at the Prince of Wales Hospital. Karen, tell me, what are your current roles and how do you divide your time between research and treating patients?
Thanks, Tanya. Hello, everyone. Well, I'm a clinician scientist, so I spend a lot of my time seeing patients, trying to care for them, and also trying to understand their problems. And then the scientific part is really to try and find solutions to those problems. So yeah, it's about 50-50. It's very variable. However, it is very variable from time to time. But it's a great privilege to be able to see patients, understand their problems, and then take the problem to the lab, bring back the solutions to them.
That's good. Now, tell us about the brain and by extension, the spinal cord. Does it repair itself? I mean, our mothers have all told us that, don't go and get drunk or take drugs, or don't knock yourself in the head playing football. If you lose those brain cells, you've lost it forever. Is that true?
Well, mums are pretty much always correct, aren't they? Um, by and large, they're right. And neurogenesis does occur. In other words, regrowth of brain cells. It can occur. In adults, it's very, very slow, and it's hard to stimulate that sort of repair process in the brain. So, yeah, I would. What I would say is that you have to. Well, I certainly tell my patients that they've got to look after what they've got, and certainly try and preserve and even stimulate the brain cells that they have left, so that you can preserve as much as you can for as long as you can.
Yes, I guess it's the use it or lose it thing that people often say.
Absolutely.
And tonight's event is brought to you by the Stem Cell Foundation, as I've mentioned in the introduction. How are stem cells and stem cell science part of your work?
Well, in two ways, really. So we in the at NERA at University of New South Wales, we use stem cells as a disease model. So our patients kindly donate material, usually a skin biopsy or a blood sample, and we're able to use the IPS, induced pluripotential stem cell research technology, to create stem cells out of those cells. For those that don't know, basically, we all start off from one cell in the body when we're born, at conception, and from conception, all of our body parts are actually made from that one cell. So, researchers in Japan basically worked out how to return each part of the body, any cell in the body, to remember to become a stem cell. That process is called reprogramming, and we use that sort of technology so that we can use patient materials, build stem cells from them, and then differentiate those into neurons rather than taking neurons from the human themselves. So it's a little bit more convenient.
Oh, yeah. So I can
And
imagine that if you can take, blood cells, for example, from a blood sample or skin cells from a biopsy, is that
yes,
right that you can turn those back into or reprogram them back into stem cells and then into brain cells rather than having to open up people's heads to get a sample of their brain?
that's right. Much more palatable for everybody. And yes, amazing technology. It's amazing what can be done now with these techniques. And yes, it's transformed neuroscience research because we're able to do it in our lab, and then create those faithful models of the brain cells, so that we can help understand what disease processes are going on in our patients that we're trying to cure or treat.
And see how they behave. I mean, it's quite powerful, isn't it? Because it's living cells that you're working with rather than, you know, tissues that are no longer living or different chemicals or different materials.
Yeah, absolutely. And they're a faithful model of what happens to that patient for
Mm.
on sepsis. And they look beautiful in the dish. For anybody's welcome, they're welcome to come and have a look or see some of our
Mm.
published articles. The living brain cells in a dish. Um,
Mm.
Most wonderful.
Wow. Thank you. And that's Carolyn. I'll now introduce our second speaker that we're going to be hearing from tonight. Professor Claire Parrish is a neuroscientist and interdisciplinary researcher who's recognised for her contributions to the fields of stem cell biology, neural engineering, and brain repair. She is Deputy Director and Head of the Stem Cells and Neural Development Group at the Florey. Claire, tell me more about your research focus and how you got there.
I guess coming back to the beginning, I've just always had this general passion for science and really followed what I loved pretty much from school all the way to where I am now. So I did an undergraduate degree in science and then pursued a research year in neuroscience, which I found fascinating at that point, understanding right at that 25 years ago what were some of the changes that were happening in the brain of someone with Parkinson's disease. And then I was introduced to a book at the time which talked about some of the very first stem cell transplants that were happening in Parkinson's disease patients. From there, I was sold, and here I am a further 20 years later, still doing the same areas of research.
Mm. Now, tell me more about Parkinson's disease. Like, what is it, and how
Mm.
prevalent is it?
I think the general community think of it as one disease. It really is a spectrum of disorders where there's every individual has a variety of different symptoms. They're largely classified, or it's generally classified as a movement disorder. So the patients will often experience difficulty in initiating or executing movement. There's the commonly recognised resting tremor that a patient has. But there's also a number of other symptoms associated with Parkinson's disease. One of the earliest ones that's acknowledged is the loss of smell. There's difficulty or there's sleep disturbances, some cognitive challenges involved. So really a plethora of symptoms there. What really brings an individual into the clinic is often those movement challenges that they have, and that is underpinned by the loss of a relatively small group of cells in the brain, and these are cells that produce a chemical called dopamine, and that's what controls our movement. So as these cells progressively die as we age, and also in an individual with Parkinson's disease, this is what leads to these symptoms.
Yeah, it's interesting because I think that a lot of people think of dopamine as, a happy hormone, and that it's something that you get your dopamine hit from, spending time and getting likes on social media. But it's also a neurotransmitter. Is that how it's powerful and its absence is related to Parkinson's disease?
Yes, it is a neurotransmitter, so that's how it works as a chemical in the brain, and it definitely does all those other things you described. So it is involved in our reward pathway. So all those good hits that we get, whether that's from, gambling or playing PlayStations or whatever it might be. So same drug, but working through different circuitry. So part of the circuitry can be those rewards that we get, and the other part can be how we control our movement.
And how common is it?
We sort of throw around numbers of between 1 and 2% of the population over the age of 65. Here in Australia, there's about 150,000 people living with Parkinson's disease at the moment, and we see about 50 new individuals diagnosed every day with the disease.
Yes. Is like the one I think of, being a Gen X person, is Michael J. Fox, and I think it appeared to me that he was diagnosed relatively young. Do we diagnose it when it's already advanced, or is it something that happens gradually over time? How soon is not too soon to be thinking about?
Yeah, it is Michael J. Fox, a remarkable man who has had such an impact on the research community in terms of his funding and what that's enabled us to do in the sector. He was an early onset case. Most Parkinson's disease is associated with the older population, and it actually emerges when we've lost the vast majority of the dopamine cells in the brain. So it's estimated that when around about 80% of the neurons die in the brain within this discrete area, that's when a patient typically really starts to have these challenges with their movement, and that's what brings them into the clinic. And we often say that the average age of onset is around about 65 years.
Okay. Yeah. And what are the current treatments, and are they good? Are there pros and cons?
So the main therapy, and Caroline might be better at this one than myself as the clinician, main therapy at the moment is really drug-based therapy, and really, like I described, the loss of these dopamine cells and the chemical they produce. The mainstay, at least treatment for those symptoms, is replacing that dopamine drug into the body. So we can do that at the moment relatively effectively in the early stages of the disease. We do know there's a number of side effects of these drugs. We know they have waning efficacy over time. That has surprisingly been the mainstay treatment for the last 60 years. There are new therapies being developed. Or not so new, there's deep brain stimulation. So electrodes put into the brain that can stimulate areas of the brain to overcome the loss of dopamine. And of course, the area of research we're interested in, which is still in its clinical testing phase, and that is looking at introducing cell therapies. We know there's work into gene therapy, which is more Caroline's research area as well. So many new things evolving. Yeah.
And preview of coming attractions, we'll be diving into both of those a bit later. But first, we're going to introduce our third speaker. Professor Kayleen Young is a neuroscientist investigating the behaviour and function of brain stem cells in order to find ways to influence their activity to achieve brain repair. Kayleen is theme leader for brain health research at the Menzies Institute for Medical Research, and she leads the NHMRC Centre of Excellence in Multiple Sclerosis Research Translation. Kayleen was one of 2 inaugural winners of the Stem Cell Foundation's Metcalf Prize for Stem Cell Research, which is part of how the foundation spends funds donated by giving grants to emerging researchers like Kayleen. It was 2014 when she won a prize, and I'm saying the royal we on behalf of the foundation. We really feel like we've backed a winner with you, Kaylee. So tell us about your research and how you got there, and what your current research is. What degree did you start with, and how did you end up here now?
Yeah, so I'm a neuroscientist and I'm working in the field of multiple sclerosis primarily. I started out doing a Bachelor of Science degree and went on to do my PhD. And one of the things I think I've loved about my science career is it's also given me the opportunity to work overseas and then to return to Australia to continue to carry out my research.
And so how did you, like, what attracted you to multiple sclerosis research specifically?
So in multiple sclerosis, brain cells called oligodendrocytes are attacked and they die. Now, these are the cells that insulate the nerves. So they wrap up and insulate the nerves in the brain, and they allow the electrical signals to be quickly and faithfully carried around the nervous system.
Hmm.
When I was working in London, I made a discovery that there are cells in the brain that are immature cells, and they're called oligodendrocyte progenitor cells. We call them OPCs for short, but they're everywhere in the brain. And what they naturally do is make new oligodendrocytes. And so one of the key things that I've been doing now for the last 15 years is trying to find ways that we can target these cells to help them make more new oligodendrocytes for repair. And a natural I guess, extension of where that research leads is potential treatments for multiple sclerosis.
Tell me a bit more about multiple sclerosis. We've heard from Claire more about Parkinson's disease. How about MS? How many people have it, and what's it like? How does it present?
Yeah, so MS is an autoimmune and a neurodegenerative disease. It affects about 1 to 2 people in every 1,000, depending on where you live. And it manifests in a number of different ways. So people might initially go to their doctor because they're experiencing tingling, they're having problems with their eyesight, they're experiencing movement problems. And all of these symptoms stem from that idea that the insulation is being removed from the nerves. And so they're not getting the electrical signals moving around the nervous system as reliably and as quickly as they should be.
Yeah. And so are there different forms, different types, or different stages? How does it present, and what is life like? And I'll acknowledge that we have Jessica has posted in the comments that she has MS. So we have people with lived experience in the audience. Tell us about that from your perspective as a researcher. What have you learnt about how it affects people's life, and what are the current treatments as well?
Yeah, so our research program works really closely with people with MS. We actually have them as part of our broader research team. We have a consumer and community reference committee that we interact with to help inform and direct our research program. And in terms of how it progresses, multiple sclerosis can start with what we call relapsing onset disease. So people might get an acute attack of clinical symptoms that might then resolve over time and then recur and then resolve. But usually what will happen after that A point is that it starts to what we call progress. And that means the disability starts to get progressively worse over time. That's probably about what is that, like 85% of people with MS will start off with that disease course. About 15% of people diagnosed with MS will start to have that progressive disease from the very beginning. And it's the progressive disease at the moment that we currently don't have any treatments to tackle.
Yeah. We'll get on to the work that you're working on, but that's a really good introduction. And we've already got a question there from Jessica about how how she joins the reference group. We'll get to those later, and I'm pleased to see questions coming through already. We will get to them a little bit later, but first off, I'm going to have a group discussion with all 4 of our speakers who will now join me on screen. And you might find that some of the questions you have in mind might come up in our discussion, but if not, do put them in the chat box. And after we've covered off the main areas that we want to talk about in tonight's webinar, we'll definitely bring in the questions from the audience. So I wanted to start by looking into the idea that I think a lot of people, when we talk about the hype and hope of stem cell science, it was originally thought that we're going to use stem cells to replace damaged cells, that they're going to be a therapeutic product or a therapeutic material. Carolyn, stem cell therapies, they've been used for 30 years. They've got an established history for the treatment of things like leukemia and other blood cancers. In your work, are there any current uses of stem cell therapies or cell therapies for brain conditions?
Absolutely. For Parkinson's disease, stem cell therapies have been used for, been trialed for the last, 20, 30 years, I think. Those stem cells, the origins are different, but basically are transplanted into the brain to replace the cells. So earlier, Claire and yourself talked about treatments for Parkinson's disease, which were symptomatic treatments, again, replacing what the cells had lost. But stem cell therapies basically try and replace the cells that have been lost in the brain. And there's been lots of different twists and turns in terms of the protocols that are being developed. But essentially, now embryonic stem cells or induced pluripotential stem cells that are differentiated into neuroprecursor lines are currently in clinical trials. And we're even about to start a stem cell trial at my institution for people with Parkinson's. So that's the first thing. I also wanted to just...
I'll go on.
I was
going to ask. You go.
I was just going to shout out to MS, not that I'm an MS specialist, but but stem cell therapies for, you talked about stem cell treatments for leukemia, but stem cell transplants, in other words, bone marrow transplants have been used for multiple sclerosis. Really, as the disease is, part of the disease is really about the immune system attacking the myelin that Kayleen spoke about. So trying to reset the immune system with the bone marrow transplant has been a treatment that has been used to treat that disorder as well. But my experience is more with Parkinson's disease. So
yeah, you were saying before about clinical trials. So in relation to Parkinson's, so that's more, it's not the standard general care, is that correct? Not yet?
At this stage, it's still in clinical trial, and although there are centres around the world that do what we refer to as medical tourism type of stem cell therapy, they haven't been regulated or been in a formal sort of situation whereby the effects are monitored. So I would encourage my patients to, if they are interested in stem cell therapy, to enter into a clinical trial where those observations are made.
And a clinical trial in Australia too, where the trial standards are high. Look, I think you mentioned there for Parkinson's, I think that's a great place to bring in Claire, because it's exactly the subject of your work, Claire. What evidence is there that stem cell therapies might work for Parkinson's? Can you tell us about that tantalizing past research, using fetal tissue grafts, and where you've got to now?
Yeah, so a bit like Kayleen described, going to do her postdoctoral studies in London, I purposely went and did my postdoctoral research over in Sweden, and that was by no accident. That is the foundation of stem cell therapies for Parkinson's disease. That's where the first preclinical studies were done. They were taking fetal tissue and transplanting it into rodent models, rats and mice models of Parkinson's disease, and able to show that they could repair the symptoms or reverse the symptoms in these animals. One thing to point out, it was realized that it very specifically had to be the transplantation of dopamine neurons, and that any nerve cell put back into the brain doesn't work. That was done around about the early 1980s, and within about a decade, they moved on to clinical trials. The reality at the time was they were using fetal tissue, human fetal tissue, which was procured through elective abortions. That comes with its own complications, of course. But we're fortunate they were able to show proof of principle that the therapy worked. And we're now in a position as a research community to see the translation of human stem cells, the stem cells Caroline described, as a more sustainable and ethical source of cells to move forward. So we now can take these embryonic stem cells or induced pluripotent stem cells. I saw a comment in the chat there. So we need to be sure that we're making that right cell type. So we are making a dopamine neuron, and very specifically of the right area of the brain, and that's what we implant back into the brain. These cells have a remarkable ability to know exactly where they need to make the right connections, and that's what restores the function.
Yes, too, that's one thing that people haven't always been aware of. It's not just the stem cell that produces the desired target cell. It's also the surroundings that you put it into. Is that right? Tell me about the importance of having the right environment and the right sort of materials, chemicals, other factors being part of the transplanted cell to make sure that it's a graft that is integrated into the person's body safely and effectively and recruited.
Yeah, absolutely. I mean, we've got to think that all these connections in the brain normally happen during development, and it's very precise about how things grow to the right place at the right time to make sure all the different circuits knit well together. When we put a transplant in the adult human brain, the environment's quite different. So normally in development, there's lots of proteins or trophic cues, we call them, that really support and nurture how these connections are established. So we can put the transplants into the brain, and we know that they have what we call an intrinsic, so an internal know-how on where they need to connect to. They don't do it as effectively as what they would do in the developing brain. So one of the research areas we've been pursuing in the lab here has been trying to stimulate that environment. And a couple of the ways we can do that is we can either introduce those proteins back into the brain. That would be akin to what would happen in development. The other thing you can do is hop on your bike and get some exercise. So we know that exercise drives some of these same proteins in the brain, and there's evidence that we can see a slowing of Parkinson's disease progression in those individuals that go dancing or do exercise. And it's much the same for these transplants. We see them knit into the brain better in the animal models by which we put the transplant in the brain and allow the animals to have access to running.
So that's animal models. What stage are we? Are we getting close to human trials, or are human trials underway? And how long do you think that this might come to the clinic as a regular standard of care in the conditions that warrant it?
So Caroline already touched on that. So we're in the position now that there's around about 11 clinical trials running globally at the moment, looking at taking these pluripotent stem cells, we refer to them as, cell products into the brain of patients. They're small trials, they're exploratory phase 1 trials at the moment to confirm the safety in particular. We've seen reports just in the last month of 2 of these trials reporting that they were both safe and they actually showed evidence of efficacy. So the patients reported benefit, and various tests conducted showed that they were showing benefit from the therapies. There's future trials planned in the foreseeable couple of years. These are going to be orders of magnitude larger in the number of patients they recruit, but we are still in that. I think this is an important thing to stress here tonight, is it's important that we take it cautiously as we move new therapies forward. We need to be sure that these are of course safe and efficacious, and we understand how they work and that they're well regulated, and we select the right patients before we move this into a mainstream therapy. I know there's a lot of hope and desire out there in the community. If things move too quickly, we can actually do more damage than good in the long run. So we're moving as fast as we can as the scientists and the clinicians, but I think we have to take these things into consideration.
I might bring you in there, Carolyn, to explain that when we say things can go wrong, I often explain to people that the difference between a stem cell and a cancer cell, both are capable of reproducing, infinitely, but one follows the plan and the other one doesn't. Is that where that risk is coming from? Can you tell us a bit more about why we need to be cautious with stem cell transplantation?
Yeah, I mean, the theoretical risk is because the cells are pluripotent that they can develop incorrectly, as you say. But now I think the literature and the field has actually progressed such that we now know that by transplanting neuroprecursor cell lines, that those risks are actually minimized. So the major risks really are about rejection of the cells. Because they are in a foreign environment, they've been sort of untimely placed, I guess, inside the brain, which is a foreign environment in some ways, despite the intrinsic capabilities of those pluripotential stem cells or the neuroprecursor cell lines. So the major challenges at the moment are survival of the cells. So in the iPS transplant program that was just recently published in Nature, I think the cell survival rate was quoted to be about 5% of those cells that were transplanted were surviving. And so there's a minimal effect. We do our clinical trial on stem cells, and this is part of an international trial. The challenges will be about the immunosuppression that will be required to allow those stem cells, or let's say neuroprecursor cell lines, survive longer. So we're over the challenges of tumor formation, although one always needs to keep that in mind. But the next challenges are really about rejection and efficacy.
Yes, yeah. And that's actually, it's the question, that's partly one that Faye B asked earlier. She also asked about the follow-ups and checks that are done post-transplantation. I guess that depends on the particular transplantation, but it would be all within the normal standard of care for any operation. Can you tell us a bit more about that?
So yeah, definitely in the protocols and a clinical trial for stem cell therapy, you're going to become friends with the investigators because it is a situation where you're closely monitored. This is this is important for the success of the trial and also for the safety of the patient. So yeah, you return frequently and have things monitored because we are looking to observe how those cells are settling in, how they're acting, and to make sure that there's no adverse events that can occur. Because often we can treat those, and the earlier we treat those problems, the easier and more minimized they are. So close monitoring. It's one of the benefits of being in a clinical trial, so that you're aware that you're being closely monitored and that your safety's center of the whole project.
And you're helping scientific research move forward. Chuan Yu has asked, hi Claire, may I know which administration route for stem cells implantation? So in a specific case of the trials that you've been involved with or aware of, how do they get to the right place in the brain?
It's all directly into the brain. It's very precise injections of cells within a millimeter degree of accuracy. So it is actually brain surgery. It is a frame you've probably seen the photos of this sort of thing. Often an individual's head is shaved, a frame is put on it, and a small hole is placed in the skull by which a fine needle then passes these cells into very precise locations. We know we don't get a new dopamine cell inside the brain through any other means. We cannot inject them through the bloodstream. We can't put them in the spinal fluid. It has to be a direct injection into the right part of the brain.
Yeah, okay. Look, I might transition over to looking at MS. Kayleen, in your work, you're looking at stimulating the resident stem cells that are in the brain, whereas the new cells are being introduced in Claire's work. So these cells that you're introducing to the brain, they produce the insulation that wraps around nerve fibers. How are you stimulating those resident stem cells?
Yeah, so I mentioned the cells, and I called them OPCs. These cells
Yes.
actually, they're touching the nerve cells and they're sensing the level of activity of the nerve cells and what's happening in the brain. And so what we've been doing is we've been using a non-invasive approach, which is called magnetic brain stimulation, or it could be called transcranial magnetic stimulation. And we put a magnet over the head of the person, and we deliver, like I guess the current goes through the coil and it creates a magnetic field over the brain that induces or modifies the activity of the brain cells. And this is picked up by the OPCs. They respond and go on to make more new oligodendrocytes. Now, we know that in the lab, this is working really nicely. We've taken it through to phase 1 clinical trials and found that it was safe for people with MS. And we've just finished our phase 2 trial, which is the one where we're trying to figure out if it's actually making a difference, helping repair brain lesions. And I don't have the results for you yet. We have finished the trial, we're analysing the data, and we will make that publicly available really soon.
I think the benefit of that is that it is non-invasive. That's very appealing, isn't it?
It is, and I'm also working, I guess, as part of a larger team across Australia with MS Australia. They've created the Australian MS clinical trial platform, and
Wow.
we're about to have a trial starting that's part of an international trial, and it's called Platypus. And it will have multiple different drug treatments that are again trying to target those cells that are in our brain, the OPCs, to help them make more new oligodendrocytes for replacement. And so more information about that is available on the MS Australia website for people that might be interested in registering for that trial going forward.
Yeah. Good. Thank you. Carolyn, I wanted to ask you, are there other ways that we can stimulate the resident stem cells in our brains to promote repair or to delay the onset of disease and aging? And so this might be, other medical interventions like the trial that Kayleen's working on, or are there other simple things like more exercise? What do you know of other brain conditions that we can, or other ways we can stimulate the stem cells we do have in the brain?
Well, Claire's already mentioned exercise, and this is basically a panacea for many things. So that needs to be done, and I can let you know that I'm a preacher. I practice what I preach, and I preach what I practice. So I've just finished my exercise session for the day. So I think that is really important in terms of trying to stimulate or to help stem cells. A number of things have been done, including using gene therapies, which can provide growth factors for these things. Electrical stimulation has been thought of. But really at the moment, the major challenge is actually to get the protocols and the immunosuppression protocols correct to allow those cell survivals. In terms of treatment for Parkinson's disease, for example, there are many different therapies, which I'm happy to talk about if you wish, but deep brain stimulation is one, focused ultrasound is another, gene therapies, which we'll later.
Yeah. One thing I did want to ask too, either yourself, Carolyn, or Claire, you might want to jump in. What other conditions might be amenable to stem cell therapy? So, for example, Raju has asked if stem cells can repair cells damaged through traumatic brain injury. Claire, I know that you work on Huntington's disease, MND, and stroke as well. Do either of you have any thoughts or comments you can make on that, but also traumatic brain injury?
Yeah, so I think we have to think about it at the moment, at least where we stand. We can replace discrete pockets of cells in the brain. I think where we start dealing with injuries that affect large diffuse areas like Alzheimer's disease or perhaps traumatic brain injury or some strokes where there's large parts of the brain involved, to be able to get cells in to replace those cells that die through the process is really a challenge. We can deliver stem cells to some degree to try and protect the remaining cells in an environment, from ongoing death processes. But in the context of stroke, we have had some success in the preclinical setting where we can model a stroke in an animal, and then we can implant new cells in, and we can show an animal that may have akin to the human condition where there's loss of a given limb, we can actually restore the function of that limb in an animal through a transplant. And a bit like we talked about needing a dopamine cell for Parkinson's disease, we have to get the right cell into the right place. So at the moment, when we're dealing with our stroke models in the lab, we're talking about an area of the brain called the cortex, or the motor cortex, that's responsible for movement. And in the dish, we now make motor cortex cells instead of making dopamine cells, and we now implant these directly back into the brain.
Mm.
Same concepts, just different disease, different part of the brain.
I guess that's the exciting part of the research, is if you can do proof of concept in one area, you might be able to roll it out elsewhere. Look, I want to turn now to the idea of stem cells as a tool, a research tool, not just as the therapy itself. So we talked earlier about induced pluripotent stem cells, where we take a patient's skin cells and turn them into stem cells and then into brain cells to study diseases and to understand those diseases better, or to test other treatments. Carolyn, can you tell me a bit about how that's part of your work?
Yeah, thanks, Tanya. We've been doing this for over 10 years now. I can't remember exactly how long. But essentially, as I said, you can take any cell from a patient and then study their disease in the dish by going through this reprogramming process and then differentiating into the neurons, brain cells that are affected. So in Parkinson's disease, we differentiate into dopaminergic neurons. For other conditions, we differentiate into cortical neurons. And we've been doing this to actually understand what happens inside those neurons so that we can understand what we need to fix up. It's actually, with scientific methods, you can actually look inside the cells. It's amazing. I always like thinking it's like Atlantis. So you can dive into the cells and see what's going on. And we fluorescently tag multiple parts within the cell. Mitochondria are my favorite parts, but other neuronal path components such as microtubules and lysosomes we can look at as well, so we can see what's actually going wrong. And just like you might want to look under the hood of your car to fix it, that's the same as you can do with
And
that.
just to clarify, because that the neurons you've created in the dish, they've ultimately come from a patient biopsy. So those cells have that patient's unique genetic fingerprint. And so if the disease has a genetic element, that's really valuable, isn't it? Is that correct?
That's correct. Yeah. No, I'm a neurogeneticist, so we see patients with genetic illnesses that impact on the nervous system, and so we take a pure approach, I guess, where we can understand the effects of that genetic mutation on the brain cells by taking a sample from a patient that carries that genetic mutation. Of course, you can genetically engineer these things, but the advantage of taking it from a patient is that they have the genetic background and context by which they are expressing the disease. The other good thing that you can do in the lab is you can also stress the cell. So, as Claire said earlier, Parkinson's usually presents when you're 60, 65. That's a really long experiment. So we try and accelerate that process in the dish. So that we can find the results out a bit faster, so for our patients.
But you can also, like, you can stress those cells in the dish, but you can also treat them as well. Tell me a bit about the role of stem cell, produced tissues or organoids in the dish that can be used to test drugs and see if they work.
Yeah, no, this is part of our program. We've been we've been understanding what has been wrong with the neurons, and we've actually used either targeted therapies, so knowing that some drugs fix up certain conditions, and then using them, or we've been doing drug discovery. So drug libraries now are available. We know what's wrong with the cells. We can then expose the neurons to drugs and see whether that problem is fixed up by the exposure to that drug library, and then we then can take it to clinical trial in into patients.
I think that's one of the things that the public doesn't have a broad understanding of is drug screening, is that there is this, like you said, a library of drugs that have been tested for safety. So they've done that first part of the clinical trial process. So we can then fast track them for the treatment of other conditions if they have similarities to the conditions that they're originally produced for. So I think that's quite, it's exciting work. Kayleen, you're working on MS, which also can have a genetic element, and you use stem cells as a way to create the disease in a dish. Tell us about the MS STEM Biobank and the MS family study work that you're doing at the Menzies Institute.
Yeah, so our MS STEM Biobank has induced pluripotent stem cells that were created from the blood of about 100 people. And this includes people with and without MS, but it also includes stem cells that we've created from members of high incidence MS families. So I mentioned that people usually, the rate of MS is about 1 to 2 in 1,000 people, but that's not true for everybody. So for some families, we do have a situation where you end up with, 3 or 4 people in 2 generations of a family that have got multiple sclerosis. And this suggests that they've got a high level of genetics that is influencing them getting their multiple sclerosis. So it allows us to put 2 things together. We can actually study the genetics of the family, and then we have the stem cells that have that same genetics. So we can create the brain cells from those people. And because of current gene editing technologies, we can actually then look at the specific contributions of different genetics of the families to figure out what it is that is happening to affect the way the brain cells work, and then how that impacts on their MS disease severity.
Yeah. And so these different approaches that you're doing, how do they combine? So I know that ultimately your aim is that multiple sclerosis can become a disease without disability. And I noted that in the chat box, one of the people in the audience said that they've had it for many years and are doing well right now. What do you mean by a disease without disability?
Yeah, so people with MS, different people will progress at different rates. Some people will find that currently available treatments help them manage their MS really well, but that's not true for everybody. And so disability is caused by the loss of nerve cells over time in people with MS. So you lose your oligodendrocytes, the nerve cells become vulnerable, and then the nerve cells die. What we're trying to do with our MS STEM Biobank and family study is find ways, I guess, really trying to find ways that we can protect those brain cells, and also then develop through drug screening ways that we can then treat people to promote and protect the brain cells, but also repair them. And in doing that, we're hoping then we can overcome that nerve cell loss part of MS that's currently not treated, and ultimately make MS a disease without disability.
It's promising. And I gather that there's resources available for people who are concerned about MS, is that right?
So on our MS Research flagship website at the University of Tasmania, we have more information about the MS STEM Biobank and other particular resources, including access to courses that we've developed that are, in particular, one that's called Understanding MS. It's a massive open online course. It's free, it's available to the public, and it involves information being provided by researchers, clinicians, people with MS, talking about their process of diagnosis. And it's about making sure that members of the public can access information that has been fact-checked and that is actually provided in a way that we hope, and the testimonies of people that have gone through the course suggest is quite engaging.
Yeah, oh, that's good. And so Kieran has posted the link to the MS Research flagship in the chat. So if you don't have the chat box, I'll read it out. It's MS Research Flagship, all lowercase, all one word, .org.au. Jessica has asked about your earlier research that you were saying that you're going to be publishing soon about your magnetic therapy trial. how can we find the results when they are released? Will they be published on the MS Research flagship website?
Yes, so the outcome of that will go on the website. It will also go out people can also sign up for our newsletter, and we then provide regular updates on those sorts of research outcomes for MS. And so, yes, we will be letting people know. But like everything that goes through the laboratory into the clinic, depending on the outcome, we may have to go back to the laboratory for a little bit and then back to the clinic again, or change the protocol. So we're hoping that it will, show some sort of improvement in terms of lesion size. But if not, that doesn't mean it's the end of the road. It means it's part of the process. So I'd like to just take the opportunity to thank everybody that's been participating in this trial.
Mm. Jessica's asking you still join the trial for it.
So for the moment, this trial is finished. We are analysing the data, but if we end up with further iterations, that information will be posted on the website as well as we look to recruit additional people going forward.
Jack has also asked how could these therapies be applied to Alzheimer's patients? I know Claire mentioned earlier that it is harder to treat brain conditions that affect larger parts of the brain rather than more localized conditions. Claire or Carolyn, did you want to make any more comment on that specific to Alzheimer's patients?
Yes, I think you're right in that it is harder to target diffuse areas of the brain with stem cell therapies, but the advances for Alzheimer's treatment are really in the biologicals. That is, having antibodies which remove the abnormal proteins from the brain. So they hold a lot of promise for people with Alzheimer's if people are wondering out there about that. So. For the first time, I feel a little bit more optimistic about treating dementia in the future, something that I'm to not have to use.
And something else, a general question, I think any of you could possibly answer this. Kev earlier asked, how do you administer stem cells to the patient? What stem cell is applied? I know this covers, many different conditions and many types, but I think it's worth just a quick explainer of the different types of stem cells and how they're used. So fat-derived stem cells or blood stem cells, which are often used in leukemia treatment, patient-derived stem cells versus ones that come from a cell line that doesn't have the patient's genetics in it. Carolyn, I think sort of as our resident clinician, I'll get you to comment on that. Can you tell us a bit about, where are stem cell transplantation cells coming from?
Yeah, well, they come from all of those sources, as you say, and they're used for different things. So for transplantation in the brain, direct transplantation, then embryonic stem cells and induced pluripotential stem cells are the ones that are currently being used. They have used bone marrow stem cells in the past, and then I think depending on what other type of disorder that you have, you might be using them, as I said, for MS. Then stem cells, bone marrow transplant cells have been used. So it depends. They can come from everywhere. Cord blood, I think, is another one. Fat cells, but they usually are. Some of those other ones are used for growth factors as much as for cell replacement. So I think that's important for the audience out there to realise that sometimes the cells are being used for cell replacement, and therefore they need to be differentiated into that specific type, or they can be used as they're mini factors, if you will, of growth hormones that can help repair organs and sometimes the brain.
Yes, I remember what Zhao Li, one of our more recent Metcalf Prize winners, she was doing research that was looking at stem cells, as she called them, biofactories that produce drugs inside an arthritic knee joint, for example, to promote repair from the growth factors rather than necessarily from cells producing new cartilage, for example. So it is interesting, isn't it? Look, that's close to all we've got time for, but I'm going to ask a couple more questions. Carolyn, you're our panel's doctor, as in the one that treats patients, not just a PhD doctor. What should we be wary of in going forward and considering different treatments? Where can we get good advice? And what general tips can you give us for looking after our precious brains?
Great question. Well, obviously, you should trust your clinician. And if there's anything that you have that you're worried about, or if you're inquiring about, then your clinician should be able to at least find out where the right information is from. We have a lot of people who have, progressive neurodegenerative diseases that are vulnerable to medical tourism, let's say, and using stem cells as a treatment when when the technology or the methodologies are not really, understood. And so that's something that you should be wary of. So your clinician would be able to either find out that information for you or know it yourself. So I think they're the take-home messages. But just be aware that stem cell therapies and the derivatives that we've discussed, we're on the next wave of those being available to treat patients and their diseases. And so, the future holds a lot of promise from that point of view.
Yeah. And I'll note that I noticed that when we were having people acknowledging country that they're coming from, there are a few people joining us from the United States. So, hello to the US. Thank you for joining us. But I will mention that different countries do have very different regulation programs for using stem cells. It might be different if it comes from your own body, say fat-derived stem cells, but still that buyer beware and exercise caution and go with, reputable, trustworthy clinicians is always an important message. And I'll be sharing some resources later that are more Australian specific, but I'll mention that in the United States, there's the New York Stem Cell Foundation. There's also the International Society for Stem Cell Research. They have some patient resources as well that have some good user-friendly patient information that has a good starting point if you want some evidence-based guidance. Finally, for all of our speakers, what are your parting thoughts or words of wisdom, any take-home message you want our audience to go home with today? I'll start with you, Claire, then Kayleen, and then come back to Carolyn. Claire.
Yeah, probably just a little bit to reiterate Carolyn's point, and that is the concept of stem cell tourism. So we have come leaps and bounds, particularly in the last 20 years, I think, since the discovery of this pluripotent population of stem cells that enable us to make pretty much any cell type in the body and treat a plethora of different disorders. But it's got to be done with caution, and the temptation to look offshore for some of these therapies that are poorly regulated is of concern. People will take your money. All we can stress is much like you're saying, do the homework, reach out to the scientists. We're very happy to give advice on these things before you embark on anything you see online.
Thanks, Claire. Kayleen.
Yeah, I think that it's just, I would like people to consider that there are different uses for stem cells and different types of stem cells. And so for some diseases, it is very much a focus on, in terms of therapies, on transplantation. But for other conditions, that's not necessarily going to be the thing that is going to be the best pathway to fix things. So sometimes we are talking then about, disease in a dish model, using stem cells for modeling. Other times we're talking about using them as vehicles to add growth factors or to use the stem cells that are already in our brain to try and promote repair. So it isn't a one size fits all. We are coming at this from multiple angles.
It's a good message, and I remember talking to one patient advocate who she had cashed in her superannuation to go to overseas on stem cell tourism and get treatment for her MS, and it didn't make any difference, and she'd lost many thousands of dollars from her superannuation, but she had a very positive outlook, and she was saying that there's so much to learn from living well with MS, from how you can change your diet, different exercises that you can do, and that sometimes the most dramatic intervention isn't necessarily the best. that probably leads me nicely to Carolyn as our clinician. What's your parting word of wisdom?
Oh, thanks. Well, I mean, now that we've expressed caution, I think I'm going to finish on an optimistic sort of note then, because I just want to remind everybody that, stem cell research has really contributed to our understanding and knowledge of neurological diseases. And we have made, major advances over the last 30, 40 years. And I'm looking forward for stem cell research to actually continue to provide us with insights and promise for new therapies in the future. So perhaps we can finish on a note of optimism because they are a very, very useful technique to not only study disease, but also to treat the disorders that we're looking at.
Absolutely. Yeah, it is an exciting field of research. And I think that given that you're looking at the genetics as well as using stem cells as a tool, and we have a lot of supercomputing, that capacity to take the information that's produced by scientists that comes from sequencing, the genome of patients so that we can have tailored medicine. I think these things are just going to change medicine as we go into the future. Well, that's all we have time for. Thank you to the audience for your questions. And please join me in thanking our panelists, Carolyn Sue, Claire Parrish, and Kayleen Young. There's a little applause thing that you can do on your Zoom chat if you like, or pop your thoughts in the comments. If you or someone you know has questions about the stem cell treatments that are currently available, we do have an information resource to help. The Foundation Guidebook, What You Need to Know About Stem Cell Therapies, can be downloaded from our website, which is at www.stemcellfoundation.net.au. And you'll be able to see the direct link from the chat that Kieran's put there, which you can see a nice preview image of that handbook, so you can download that there. And today is the 5th of June. What a coincidence. It's nearly the end of financial year. This is the time of year when I say, if you'd like to make a tax-deductible donation to the foundation, that would be lovely. You can do so via www.stemcellfoundation.net.au/donate. We use funds donated by the public to provide patient and community education, such as events like tonight's webinar. Our next event will be an in-person event in the Gold Coast in November. And if you'd like to hear about events and stem cell news in general, sign up for our newsletter via our website. You'll also see links there to past webinars. So for example, at this time last year, we had a webinar that looked at stem cells and women's health, looking at endometriosis and pelvic organ prolapse and breast cancer. So there's lots of useful information that you can find there. But the vast bulk of the funds that are donated to us go to supporting promising Australian researchers, such as Kayleen as an inaugural Metcalf Prize winner. The scientists we've funded are working on everything from basic fundamental research to improve treatments for cancer, MS, cystic fibrosis, heart disease, diabetes, vision loss, osteoarthritis, and many more. And you can read about these researchers we've supported on our website. If you'd like more information about making a donation, you can email our General Manager, Graham Megan, via inquiries@stemcellfoundation.net.au. Thank you again for joining us tonight. We hope to see you at our future events, but in the meantime, we hope you have a great evening. Thank you.